So thus, over a period of time, due to secondary growth, lot of changes take place in the plant body. As the vascular cambium becomes active and additional tissues are being added, there will be a kind of pushing force getting created within the plant. As the stem continues to increase in its breadth due to the activity of the vascular cambium, the outer cortical and epidermal layers, they get broken and they need to be replaced to provide new protective cell layers. Hence, sooner or later, another meristematic tissue called cork cambium or phellogen develops, usually in the cortical region and in the dicots. The collenchyma cells which form the hypodermis contribute to the formation of this secondary meristem called cork cambium or phellogen by dedifferentiation. Thus, collenchyma are unique because they not only provide mechanical strength and support, because they are living, they can even perform photosynthesis, and under crisis situation, they may also resort to cell division by dedifferentiation. The phellogen that develops at the cortical region, it starts cutting off new cells on both the sides. The new cells that are produced towards the inner side develop into secondary cortex or phelloderm. The phelloderm is made up of thin-walled cells. The new cells that are produced towards the outer side give rise to what is called cork or phellum. In fact, the cork or phellum consists of thick-walled, suberized dead cells. They are impervious to water. And these three layers together constitute what is called periderm. The periderm thus consists of, from the outer periphery, the phellum or cork, middle phellogen, and the innermost phelloderm or secondary cortex. In some of the dicot stems, we do find at the extreme periphery of the plant stem a band of dry, hard portion. This constitutes what is called the bark. The bark consists of all tissues lying outside the vascular cambium. The bark that is formed early in the season is called early or soft bark. Towards the end of the season, late or hard bark is formed. In fact, in the family Myrtaceae, exemplified by plants like eucalyptus and guava, over the stem you can see a thin membranous hard layer peeling out regularly. This is the thin bark which is characteristic of the family itself. As a result of this secondary growth, in many of the dicot stem, small lens-like openings are produced at the epidermal area through which water can easily be lost by the plant, and these lens-shaped slit-like openings are called lenticels. In fact, the formation of the cork and the secondary cortex by the phellogen is mainly with the intention of preventing this loss. If we look at the lenticel area, we find below these small slit-like openings loosely arranged cells called complementary cells, and these complementary cells cover the phellogen as well as the secondary cortex and the interior of the entire plant, so that the loss of water is minimized to a greater extent. However, as these openings are present in old dicot stems, very low quantity of water gets lost by the plant in the form of vapor, and that itself we call as what is called lenticular transpiration. Although these openings are present and account for the loss of water in minimum quantity, they play a very important role as they facilitate easy exchange of gases between the environment and the plant body.